So today we are going to talk about carbohydrates. Okay? Yeah, we finished water, we finished those mineral ions. In other words, we finished the inorganic nutrients. Okay? So today we are going to begin with organic nutrients. Okay? Those ones which we are studying organic chemistry. Those elements of carbon and hydrogen. Okay? Or enemy compounds of carbon except carbonates, oxides, and hydrogen carbonates. So also these carbohydrates, the organic nutrients, because they have carbon directly bonded to hydrogen. Okay? Yes, I know this might not be new. I know you've ever heard of this. Because in senior 2 you studied this. Okay? However, now we are going to add knowledge from where you stopped from. That's where we are going to begin from today. So we are going to add on knowledge. So carbohydrates, they are organic nutrients, the organic compounds made up of carbon, hydrogen, and oxygen. Okay? Carbon, hydrogen, and oxygen only. No other element is involved. Okay? And they have a general formula, okay, of C5. This is an example. They have a general formula of C, okay, Cn H2 water O and then Y. Or you can say C CH2O. So whether you write this, it all remains the same thing. Okay? So this is their general formula. In other words, all carbohydrates, they conform to this general formula. Okay? Where N is the number of carbon atoms. Whereby, if you have 3 carbon atoms, then you are C3, H what? H6, and then O3. And this one is a triose sugar. Triose sugar. E.g. dihydroxyacetone and then the glyceraldehyde. These ones, they are triose sugars and they have the general formula of C3H. We are going to see each. If N is 4, if N is 5, then you have C5, okay, H10, and then O5. And this one is a pentose sugar. And examples of a pentose sugar, we have two. One is deoxyribose sugar, and then another one is a ribose sugar. We shall study each, okay? And we shall see how they behave and what are they. If N is 6, maybe let me write it here. If N is 6, then this one becomes C6. Can you guess? H12. Or 6. Is that good? And so from this one is a hexose sugar. You know hex. Carbohydrates are named by adding suffix "-ose". We are going to talk about all of this, so have no worries. So this is a hexose sugar, and an example we have glucose. We have fructose. We have galactose, and so on. Is there any other? Yes. So depending on the number of carbon atoms, then it will determine which sugar Yeah. But all of them, they do conform to this general formula. Or you can write it as this. It is all the same thing. Is it okay to everyone? Is it still public knowledge? Yes. So we usually move from simple to complex. That's how knowledge is gained. You start with what you know, you start with known, and then you gradually move to what is not known. So those are carbohydrates for you. They all do conform to that general formula. Now carbohydrates can be sugars, okay? And with sugars we mean monosaccharides and disaccharides, or they can be complex molecules, complex sugars called polysaccharides. And you can find out the examples, okay, of these polysaccharides. And we are going to see. Now before we bother ourselves with carbohydrates, before we waste our time with these carbohydrates, you need to find out why. You need to know why we are studying these carbohydrates. Not so? Is it worth it? Or it's a wastage of time? Okay? So, I think it's really worth it. Why? Based on their function. And they have enough. Number one, they are primary source of energy. Primary source of energy. I'm not saying that they are the only source, no. But they are the primary source. Your body feels nice while metabolizing carbohydrates. If you provide carbohydrates, enough of it, and you provide proteins, enough of it, and you provide lipids, enough of them, the body is going to metabolize carbohydrates. Okay? So therefore, these carbohydrates, they are really primary sources, okay, of energy. And energy, from our P7 definition, energy is the ability to do work. Without energy, then you can't do anything. You are nothing. Okay? You are nothing. You cannot live without energy. And so these carbohydrates, they are good in that. They are the primary sources of energy. But they don't stop at just energy. Okay? They do form structures. For example, cellulose. Okay? This, the cellulose cell wall. So the cellulose, how the cell wall is important in plants, both maintaining, both on a cellular level, on a cellular, okay, on a cellular level, English, okay, and both on the organism as a whole. How the cell wall is important, and the cell wall is purely cellulose, and cellulose is purely a carbohydrate. Okay? Yes, these are glucose molecules. Glucose molecules. However, why is the cellulose, since they are all carbohydrate, why is it different from starch? They are all polymers of carbohydrates. They are all polymers of glucose molecules. Very many glucose molecules, they add up together, okay, to form a polysaccharide. But why is cellulose? Think about that. Why is cellulose different from starch? Yet they are all made up of glucose molecules. And even why should they be different? Because they all do perform different functions, right? Yes, this one storage compound, but this one structures. So they must have different functions. Sorry, they must have different structures. Okay? And so can you really account for the difference in structures? Can you? If you can do that, then you are good. But if you can't, then that's what we shall talk about. by the end of this lesson, actually not today's lesson, but by the end of carbohydrates, then we shall be able to know the structure of glucose, sorry, the structure of cellulose and the structure of starch, how the two are formed, okay, and why the two have different structures, by the way, okay, and what makes them have different structures. Okay, that's brilliant. That's brilliant. That's brilliant. So carbohydrates don't stop at that. Okay, but they also determine the osmotic pressure of body fluids such as blood. For example, in homeostasis, you studied that glucose levels must be maintained constant. So why should they be maintained constant? Why should they be maintained constant? Okay? Otherwise, why shouldn't they be high? Why shouldn't they be low? Why should you just leave them to vary the way they are, the way they want? Okay? So they do contribute a lot, okay, to maintaining the osmotic pressure. And you know, if you maintain the osmotic pressure, you know how good it is. Hmm? If if the pressure of blood increases, if the osmotic pressure of blood, for instance, increases, then blood is going to take in more water. So the blood volume increases so high than what is expected. And so you know the effects, what will happen to the heart, what will happen to the blood vessels. Too much force will be created, which will burden, and so issues. Similarly, when it goes below, okay? So therefore, you must be able to regulate the blood volumes and blood osmotic pressure. Okay? That's brilliant. We've studied cell biology and we've talked about structures like glycoproteins and glycolipids. They are so important. Okay? They are sites for recognition. Okay? They are sites for recognition where enzymes, where hormones they will bind, antibodies, that's where they will bind. Cells communicate with other cells, that's where they will bind. But they are all carbohydrates, short segments, short chains of carbohydrates. We can call them oligosaccharides that are bound to the protein, bound to the lipid. Okay? So without these carbohydrates, then I don't know if you'd be able to communicate within your cells. So those are the carbohydrates for you, and I hope now you know why we should study carbohydrates. Okay? And they are a good number of functions. Okay? We have a good number of functions, even beyond what we have just listed. So those are the carbohydrates. Now, they do occur in types, and I know you've ever heard of these. And the types include monosaccharides, disaccharides, and polysaccharides. But meanwhile, let's not take things obvious. Now, mono means one, okay? One, and saccharide means sugar, okay? So if you want to be more fancy, you can also say one sugar, monosaccharide. Then di means two, and so this is two sugars. And then poly means many, okay? And so you can refer to this as many sugars. So now therefore we classify carbohydrates basing on the sugars that they have. If you are just made up of just one sugar, okay, just one sugar, one hexose sugar, oh, this is wrong. Now I'm drawing benzene instead of carbohydrate. This is 1, 2, 3, 4, 5. No, there is another one here. So this is 1, 2, 3, 4, 5. and then 6 here. This is O. So if I just draw this, and it is just one sugar, okay, then that one is monosaccharide. It's a monosaccharide. But however, when the two sugars, okay, join together, okay, maybe you have this and that's one sugar, okay, and then this is your O, and then your one sugar joins to another sugar, okay, something of this kind. Your one sugar joins another sugar to form such a compound, then this one is a disaccharide. Okay, but if very many sugars condense, very many, okay, very many, okay, then they form what we call a polysaccharide. Okay, polysaccharide. Now as you are reading, there is this word which you may find. Which word? This one. Why should I write it here? Oligo. You may find this word, oligosaccharide. Okay, the rest is the same. Now this one, it refers to a carbohydrate with few sugar molecules. It is not a disaccharide, neither a polysaccharide, but it ranges from 3 to around 8 sugar molecules. So this one, it is an oligosaccharide. Okay? Yes, however, they are rare. You might not find them. Now let's talk about monosaccharides. Oh, sorry. So let's talk about monosaccharides, and simply mono, as I said, mono means one and saccharide means sugar, so it is one sugar. So this is a carbohydrate that consists of just one molecule of sugar, one sugar molecule. So they are usually referred to as simple sugars, and they have different properties. Now, if a question says properties of sugars, properties of monosaccharides, properties of disaccharide, think about a monosaccharide such as glucose, and none other than glucose, but the glucose that you buy from the shop. Okay, now this glucose, is it sweet? Yes, it is sweet, that's why you buy. Does it dissolve in water? Yes, it dissolves. That's why when you put it on the tongue and you produce saliva, it will dissolve. Is it a powder or they are crystals? Okay, they are crystals. Okay, now already you have three properties. Sweet, it is soluble, and it is crystalline. So you already have three. Now after these ones, then you think about the rest. So properties, they have a sweet taste. All monosaccharides, they are sweet. They all do dissolve in the water, and they all form crystals. They are crystalline, not powder. Then they have the lowest molecular mass compared to others. The disaccharides, the polysaccharides, and the oligosaccharides. These ones have the lowest molecular mass. And finally, they can pass through a selectively permeable membrane. You know a whisking tube in olive. So if you get your whisking tube, okay, maybe this is a whisking tube, and you tie this end, you tie also here, and inside you put monosaccharides, you put disaccharides, one like this, and then you put a polysaccharide, and then you dip the whisking tube in water, for instance. Yes, water is going to enter, but also in this neighboring solution you find there a monosaccharide. Okay, which implies that these monosaccharides can truly pass through the selectively permeable membrane. A moment, So basically those are the properties of these monosaccharides. They are all sweet, they are crystalline, and they dissolve in the water, and they pass through a selectively permeable membrane. And finally, they have low molecular mass compared to others. Let's see how they are named. Okay, let's do like we studied this, we talked about this. Now when we are naming these,
Excuse me,
yes, Edwin.
don't they have effect to Benedict solution?
Yes, they do. Actually, they do. They do have an effect on the Benedict solution, whereby,
So it's a property.
yeah, it is. It can work as a property. It is a property. The property is they are reducing sugars. Okay, in that they do change the color of Benedict solution from blue solution to green solution. Okay, then to yellow PPT, then to orange PPT, finally to brown PPT, depending on the amount, okay, that one has in the solution. Somebody has joined? So yeah, they are reducing sugars, so they change the color of the Benedict's solution. They have an effect on the Benedict's solution. So let's talk about the nomenclature. Now we name these monosaccharides, even some disaccharides, okay, by adding suffix OSE, okay, OSE, or you can call it was, okay, on the stem name, okay. For example, if N is 3, okay, then that one becomes a triose sugar. If N is 5, then that one becomes a pentose sugar. For example, you have a ribose sugar. So are you saying a ribose sugar or a deoxyribose ribose sugar? So are you seeing still the OSE comes? But because it has 5 carbon atoms, then it is a ribose sugar or a deoxyribose sugar. And we are going to see
Teacher,
all.
pardon.
So we name these carbohydrates, these monosaccharides, by adding suffix OSE, okay, on the stem name. And for instance, if the stem name is gotten from the number of carbon atoms, of course, whereby if it has 3 carbon atoms, then that one is a triose sugar. First, a moment, I'll talk about this, okay. Then if it has 5 carbon atoms, then that one becomes a pentose sugar. And examples of pentose sugars, we have a ribose sugar and a deoxyribose sugar, okay. So you can see they all have the OSE, the OSE. If it has 6 carbon atoms, then that one is a hexose sugar, okay. An example, you can have your glucose. You can see glucose, still it has the OSE. Fructose, it still have the OSE. Galactose, the same. Galactose. These are examples of hexose sugars. Okay, still the same. Now, if it is 7, then that's a heptose sugar. However, this one is really not common. Okay? Yeah. Now, for triose sugars, these ones have 3 carbon atoms just. And they are classified in 2 categories. basing on the groups that one has. If you have this group in your structure, okay, if you have this group in your structure, then this is an aldose group, an aldo group, or an aldehyde group. But if you have this group in your structure, now this is organic chemistry, and we shall study all this. If you have this group in your structure, then this is an alkyl group, and this is another carbon atom. Then this one is a ketone group or a keto group. Okay? Yes. Now, if you have an aldehyde group, then you are an aldehyde. Okay? And for example, it is glyceraldehyde. It is a glycerol, but instead of a glycerol, one of the OH is replaced by this group. So it becomes glyceraldehyde. And this one is so important in respiration. Okay? For those who have studied respiration, you know how important these ones are. For those ones who haven't, we shall still see. But these ones, they are so important as far as respiration is concerned. Now, if you have a keto group, okay, then you are, for example, dihydroxy. It is di. It has two OH groups. So dihydroxy, acetone, okay, dihydroxy, acetone. Now, the same glycerol, but it has this group, okay. The second carbon atom has this keto group, and then the rest are hydroxide ions, hydroxyl ions, okay. So basing on the group that you have, then it will determine which carbohydrate is that, okay. Yes. And the most common carbohydrates, they are the monosaccharides. Sorry, yes, the most frequent monosaccharides are hexose sugars, and that is the glucose, the fructose, and the galactose. Is that okay? I hope I've explained. In case you have a query, it's okay, you can unmute and ask, or you can drop it in the chat box. I'll find it. So let's talk about our hexose sugars. So from hex, okay, like we said, hexose, these ones, they are made up of 6 carbon atoms, okay. And examples we have glucose, and then galactose and fructose. Now, all these hexose sugars, they conform, they have this general formula. The formula is the same, okay, but they have different structures. In other words, they are isomers. And good, we studied isomerism, okay. Good, we studied isomerism in chemistry, so you know what isomers are and you know how they behave, okay. They have the same molecular formula, but when it comes to the structure, then the structure becomes different, okay. So they all conform to this general formula. For instance, when you look at this, you can see you have this. Oh, let me get some different pen. Maybe this, because we already have red here, so I can't use red again. So you can see, okay, they all have 6 carbon atoms. 1, 2, 3, 4, 5, and 6, okay. So even this one, now let me first take off this. Let me use this pen. So they have 1, 2, 3, 4, 5, 6, okay. Even this one, 1, 2, 3, 4, 5, 6. So they all have the same Okay, molecular formula. However, now when it comes to the structure, now the structure becomes different. Okay, for instance, you can see this one has an aldehyde group. Okay, but you see this one. Okay, it has a keto group. This hydrogen is not there, it's a mistake. Okay, this hydrogen is not there, it's a mistake. So this one has a keto group, whereas this one has an aldehyde group.
Teacher, what is a keto group and what is a aldehyde group?
If I write C, okay, and then the rest are carbon atoms that are bonded to this C, then this one is a keto group. But if I write C with this O, but one is a carbon atom, but another one is a hydrogen, and this one becomes an aldehyde group. So if you are to see, check on this carbon atom here. It has the oxygen, okay, and the oxygen is bonded to carbon like this, but it also have this hydrogen, and this is the hydrogen. Now this is the carbon, this one. So this is an aldehyde group. But for this one, you can check on it, okay. It has this carbon. Now this hydrogen is not there, okay, because like I told you, the concept is organic chemistry, carbon must have 4. It has 1, 2, 3, 4. So this hydrogen is not there, it is a mistake. So it has this and this, and then you can see the oxygen, not so. So that is a keto group, and this is an aldehyde group. So they all have that general formula. However, they have different structures. Okay? They have different structures, and so they are referred to as isomers. Okay? For instance, glucose itself, it can exist also in different numbers. Okay? Different isomers. Okay? Whereby in the first isomer, this is carbon 1, okay? And then this is carbon 2, this is carbon 3, 4, 5, and so on. So the OH might be this side in this, but now in this side, the OH is here, and you see in carbon 3 where the OH is, and you see in carbon 3 where the OH is. Okay? But you can also see this. So you might see these differences as minor. You might see these differences as minor, but these differences, they are actually the ones that bring about the different properties. They're the ones that bring about glucose to be glucose. Okay, let me get some pen. They are the ones that make glucose to be glucose. Glucose to be glucose, galactose to be galactose, and fructose to be fructose. Yet they all have the same molecular formula. Okay? Yes. Is it okay? You are free to ask. We will just go slowly by slowly. I want to drive you speed. You can put that in Uganda. So I won't take your speed. Okay? So you may look at these differences as minor. Okay? You see here the OH on carbon 2, it's here, here, here you see it is this side. So you may look at this as just something small, but actually these differences, okay, they are the ones that bring about the differences. Glucose to be glucose, galactose to be galactose, and fructose to be fructose. Okay? Let's proceed and look at this. Now, our hexose sugars, we are studying them as linear, as you can see. Which pen is going to work here? Because as if I have, let me use red. So we've been studying them as linear molecules. But in organic chemistry, we are going to borrow some knowledge in organic chemistry. You are aware that these molecules, these organic compounds, with their linear structures, they are unstable. Okay? They are unstable. N-hexane, 1, 2, 3, 4, 5, 6. N-hexane is unstable compared to cyclohexane. Okay? N-hexane is unstable compared to cyclohexane. So as a result, also in nature, these hexose sugars of ours, the glucose, galactose, fructose, they also occur in cyclic form, in ring form. Okay? Yes. So you can see, and how is it going to ring? So if this is your ring form, let's first rub. If this is your ring form, are you seeing this OH on carbon number 5 and this group on carbon number 1? That is what is going to bring the difference. Okay? And how is it going to bring the difference? The OH, oxygen. Now this is some organic chemistry. Oxygen has some lone pairs of electrons. Okay? So,
What are lone pairs?
what are lone pairs of electrons? So if I take you back to organic chemistry, to inorganic, where we draw, I'm going to draw carbon. This is some chemistry. This is chemistry, and I'm going to but okay, let me talk about lone pairs. However, they are not needed here so much. So if carbon with 6 is going to be 1s2, 2s2, and then 2p, 2p what? 2p2. No, this won't help. Anyway, the lone pairs of electrons after bonding. Now this one is too much. There will be hybridization. So this won't help us. But after bonding, for example, oxygen, which is 2 to 6, oxygen is 8. Okay? Which is 2 to 6. After bonding, okay, it will gain 2 electrons. Okay? Now the 2 electrons to form a bond. Actually this one. Good enough, we have oxygen here. So the oxygen, it will use 2 electrons to form this bond and another bond with this hydrogen. Now since it has the 6 outermost electrons, okay, the 2 electrons, they will form a bond, then you remain with another electrons that are not involved in bonding. Okay, so out of the 6, you will use 2 and remain with the 4 electrons that are not involved in bonding. Okay, now those electrons that are not involved in bonding, yes, they occur in pairs, okay, but they are not involved in bonding, so they are the ones that we refer to as lone pairs of electrons. Now we shall study this more, we shall understand this more in chemistry, especially physical chemistry. Okay, that's when we shall talk about it. Now here, since oxygen, it has a lone pair, it will come and take away this hydrogen. Okay, it will come and take away this hydrogen. Okay, and then the remaining, okay, electrons on this, they will be donated to this carbon 1. Okay, they will be donated to this carbon 1, as you can see that illustration from here. I think so. The oxygen, it will come and take away this hydrogen. Okay, this oxygen, it will come and take away the hydrogen here. Let me rub and I repeat it. This oxygen, this one, it will come and take away the hydrogen here, okay, and then the remaining electrons, they will be donated to this carbon number 1, as you can see how it is illustrated here. So are you seeing this blue hydrogen that finally it will be on this oxygen? Okay, yes, and you see that the hydrogen is remaining, and you see that the oxygen is forming a bond with a carbon. Yes, so after forming this, okay, then you have your final structure, okay, of glucose. Now, 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 are you seeing these OHs? Their orientation matter. These OH groups, their orientation matters a lot. Whereby,
Teacher,
yes,
teacher,
yes, Otim.
the oxygen four lone, the four lone pair, the two lone pairs.
Mm.
It donates all the two lone pairs to the
Oh, no.
hydrogen, only one lone pair
No, it donates one. It donates one lone pair, not two.
to the hydrogen.
Yeah,
Okay.
yeah. So it will donate, to come and donate one lone pair to this hydrogen, and then it goes away. Then this one takes away the electrons and donates the electrons to this carbon, like how you can see it here. Okay, and so you end up forming a ring structure, and this is a ring structure. Now note, you see the orientation of these OH groups, this group, this group, this group here. So you see the orientation of these OH groups. It's not a minor issue. It's not a minor issue. Actually, actually, the orientation of these OH groups, these ones, it's the one that creates a difference in our exhaust sugars. It's the one that brings about galactose, okay? And galactose will appear different from glucose. Even among glucose, the orientation of these OH groups, okay? It's the one that creates a difference between alpha glucose, and I don't know if you have ever heard of that, alpha glucose and beta glucose. And these two glucose molecules, they are different. And so basing on their differences, they are found in different structures, different compounds. And these different compounds, they do perform different functions. Okay? Somebody has joined. So basing on the differences that they do possess, okay? Actually, basing on this OH, it is not minor, like I told you in the above. Those different orientations, they are not minor. Here, the orientation, this OH being down and this OH being down, it can be up. Okay? The OH here can be up. The OH here can be up. This OH here, it can be inside. And this one, it can be outside. And basing on that, it creates a big difference in both the appearance and the physical property of these monosaccharides and the physical properties of the compounds that are formed from these monosaccharides. Is that okay? Is it sinking into your cranium? Yes, let it sink. Let it sink. So let's proceed. So you mark the orientation of these OH groups. It is not a minor issue. Now let's be specific. Let's now talk about the pentose sugars. And like we said, pentose, red in red. Pentose sugars, pent means 5, os, that's how we name carbohydrates. So pentose sugars, these are sugars that are made up of just 5 carbon atoms. Okay? Yes, they have 5 carbon atoms, and they are found in nature as ribose. These are the examples, the ribose and the deoxyribose sugar. Now, they do exist as straight chain, however, the ring form is more stable than a straight form. For example, let's have a look. Let's have a look at this. So you have a ribose
Teacher,
sugar. Yes.
first remove the status bar down.
It is this one. It is removed. Let me first talk to this guy. Yeah, it's okay. So I hope everything is now okay. So like I said, paint 5, then os, sugar. So it is a sugar with 5 carbon atoms. We have 2 examples that are known. We have a ribose sugar and a deoxyribose sugar. And these two have almost similar structure. However, the difference is on carbon number 2. We are going to see. So you can see the straight chain of a ribose sugar. It's like this. Okay? On carbon number 2, this is carbon 1. This is carbon number 2. Okay? The ribose sugar, it has the OH group. Okay? You can see. But the deoxyribose sugar, okay, it has, okay, even here there is an OH group. No, it is not supposed to be. It is the H group, like how you can see here. So on carbon number 2, that's where the difference is. The ribose sugar has the OH group on the carbon number 2. This is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and then this is carbon 5. Okay? So on this carbon number 2, that's where the difference is. You have there the OH group. But for the deoxyribose sugar, deoxy, it means remove oxygen. Deoxyribose, then it does not have an oxygen atom on carbon number 2. And that's where the difference is. And so these two sugars, they'll be found in totally different compounds. For instance, you find a ribose sugar in RNA, in ribonucleic acid, but you find a deoxyribose sugar in DNA. And you cannot have this in DNA. You cannot find the ribose sugar in DNA. You cannot. And you cannot find deoxyribose sugar in RNA. You cannot. But what is the difference? It is just this one minor this difference you call minor. Okay? It is the one that is bringing about that difference. So really, when we are these differences you call minor, even when you are writing isomers in organic chemistry, please take note. They are not minor differences. Okay? Those minor differences, they actually create a big impact, and that is the big impact. So one is a ribose sugar, another one is a deoxyribose sugar. And one is found in RNA, another one is found where? In the DNA. Now let's talk about the triose sugars. Triose comes from three. Tri, okay? Let me get some yellow pen. Triose sugar. Triose sugar. So this is a sugar that is made up of 3 carbon atoms. Okay? Sugar that is made up of how many? 3 carbon atoms. And we have 2 known examples. The first one is glyceraldehyde, okay? And the second one is dihydroxy acetone this is I, not D. So dihydroxy acetone. Okay? So those are 2 common examples of triose sugars. Not so? Yes. And they have the same formula. Everything is the same, but the difference is just 1. The difference is just 1. 1 like this. And you are going to see the difference. And they are found in plants and animals, and they are intermediates in respiration. Okay? Now, where in respiration, maybe let me just give you a hint. Just a hint. Okay? Then we shall see it in details. Okay? When we reach there. However, I made videos as far as respiration is concerned, because it's what I am teaching senior 6. And whenever I teach, I record. So I have videos. Maybe if you are insinuated, you can come for the videos. You can watch the video, okay, on respiration. So the glucose, you know, it is glucose which is the main metabolite, the main product, the main reactant. So you have your glucose. Now the glucose is going to be phosphorylated, okay, to form one compound. I don't want to speak the names because I don't want. Anyway, we are sciencers. So you phosphorylate glucose. When you add one phosphate group, then we shall form the glucose 6-phosphate, and then you isomerize, then you form fructose 6-phosphate, and then you phosphorylate it again, you form fructose 1,6-bisphosphate. Now that fructose 1,6-bisphosphate, it is the one that is a high energy product that you will undergo split. It will split itself into 3 carbon compounds, and the first one is glyceraldehyde 3-phosphate, and the second one is dihydroxyacetone phosphate. So these ones, you see how important these 2 are in respiration. Yes, so they are equally important. Now they are almost the same, but they have just a difference, whereby one is an aldehyde. You can see this group, okay? One is an aldehyde, but another one is a ketone. But in all, they are all formed from glycine. So if I do this, this is my glycine. We shall find it in fatty acids. We shall find it in lipids. So this is my glycine, and the rest are hydrogens. This is hydrogen, 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 hydrogen. Now if I oxidize this carbon 1, okay, it will turn into an aldehyde group, from alcohol, okay, to aldehyde, okay, and the rest are the same. As you can see, the rest are the same. And so therefore this one becomes from a glycerol now to glyceraldehyde. If you study organic chemistry, if you study ketones, it will become easy for you. That's why it's better to do biology when you know some chemistry. Okay? The two do overlap. Okay? the two work together. So when you have your glycerol, which is an alcohol with 3 OH groups, but when we oxidize this alcohol on carbon 1, we shall form an aldehyde. And it will cease from being a glycerol, just glycerol. You know how alcohols are named. We add OL, and this is an alcohol, so that's why there is OL, and it becomes glyceraldehyde, where we replace this OL with an aldehyde. Okay, so now this is our glyceraldehyde. Similarly, when we oxidize this carbon number 2, it turns into an carbonyl compound still, where this is carbon and this one is also carbon, as you can see. Okay, as you can see. So now this is glyceraldehyde and this is di. Why di? Because of 2 OH groups, this one. Okay, that's why it is dihydroxy. Then acetone, dihydroxy, acetone. Why acetone? Because of this CH2, CH2, CH2. So basically it is as simple as that. Now, the two can also exist in ring form, where that is a ring, and then this is another ring. Oh, I'm going to erase everything. Yeah, this is another ring. Now, however, you don't need to cram this is biochemistry, not biology. You don't need to cram these structures. Okay? But I need to give you a hint. I need to give you the real science, okay? But nobody is going to draw this structure and say, name, what is this structure? But you know that a glyceraldehyde is a triose sugar. Dihydroxyacetone phosphate, dihydroxyacetone is also a triose sugar. Because an objective question can come, which of the following is a triose sugar? Okay? And then they write 1, glucose, number 2, galactose, number 3, glyceraldehyde, number 4, dihydroxyacetone phosphate, and then you hustle it out. So those are the triose sugars. Okay, yes. So let's see some few importances of monosaccharides, some few importances of sugars before we proceed to real science. 1, triose sugars, they are intermediate products in respiration and photosynthesis. Okay, and other branches of carbohydrate metabolism. Like I talked about respiration, even photosynthesis, it is the same thing. Okay, we have the dark stage, okay, and then the light dependent stage. That's photosynthesis, divided into 2 stages. So whereby in the dark stage, the ribose sugar, it is not ribose sugar, but it is rubub. Okay, it is rubub. Rubub. It is ribulose biphosphate, ribulose bisphosphate. So you see, it has also a pentose sugar. Okay, now this ribulose bisphosphate, it is going to accept carbon dioxide to form a 6 sugar intermediate product, which will split to give 3 carbohydrate, or a carbohydrate with 3 carbon atoms. Okay, glyceraldehyde 3, phosphoglycerate, yeah, it is phosphoglycerate, which is then going to be reduced to form glyceraldehyde. So you can see in all, you still find the triose sugars. Okay, then the pentose sugars, you can find them in nucleic acids, that is ribose in RNA, and then deoxyribose in DNA. Then you can find them in coenzymes. For example, the ribose, we shall find them in NADP. You know this NADP? It is nicotinamide adenine dinucleotide phosphate. That's how it is filled. Nicotinamide adenine dinucleotide phosphate. So because it has nucleotides, we shall study all this and you shall see that a nucleotide has a ribose sugar. Now, depending on which nucleotide, if it is RNA, then it will have a ribose sugar.
Teacher,
Yes.
so if it has the ribose sugar, does it mean that that ribose is going to make for it the proteins?
If it has a ribose sugar, pardon the question?
You have said we shall see that it has the ribose sugar, no sir?
Yeah.
So what is the importance of that ribose sugar? Is it the same as the one in the cytology, or
Oh. Now, the ribose sugar, this one, it is just, it's a sugar. Okay? We have seen its structure. Okay? We have seen its structure. Well, this is O, and this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 here. So it is found in the nucleotides. Okay? So we use this ribose sugar, an organic base, together with the phosphoric acid to form a nucleotide. We shall reach there in nucleic acids, where we shall study DNA, we see. So a nucleotide has those three: a ribose sugar, an organic base, and phosphoric acid, or a phosphate group. So therefore, these ribose sugars, if they are not there, then you cannot form these nucleotides. Okay? And then if you can't form a nucleotide, then there is no way how you're going to have the NADP. Okay? And there is no way you can have the NAD. And there is no way you can have the FAD. I think this is FAD. Yes, it is FAD. There is no way you can have the FAD. And these coenzymes, these groups, these molecules in your body, they are important. For example, this NADP, it is the one which accepts electrons in plants during photosynthesis. In that now the electrons are carried from one area to another area where the electrons are needed. So without this NADP, okay, then there is no way how photosynthesis can occur. This NAD is the nicotinamide adenine dinucleotide. Okay? The difference here is nicotinamide adenine dinucleotide phosphate. This one is nicotinamide adenine dinucleotide. So this one does not have a phosphate group, but it also has a ribose sugar, and this one is found in animal cells where respiration takes place from. So if you don't have these electron carriers, FAD, this is flavin adenine dinucleotide. So if you don't have them, then respiration cannot occur. Photosynthesis cannot occur. Similarly, ATP, and now all of us here know what ATP does. Sincerely speaking, I'm able to teach you because I was able to respire to produce ATP so that I can be able to speak, be able to read these words, transmit and transduce the light from these words into an impulse taken to the brain, interpreted this as ATP. Able to speak. Imagine if you are speaking, the jaw keeps moving up and down, up and down, up and down, up and down. Which jaw? The lower jaw. The upper one is rigid. So, but for the lower jaw to move, okay, then the muscles must contract and relax, contract and relax, contract and relax. Now, For the muscles to contract, then ATP must be there. Now, if you don't have the ATP ready, then you are useless. Okay? Obviously, you die. ADP, same as ATP. AMP, this is adenine monophosphate. No, not adenine, but adenosine. Adenosine, meaning that there is a ribose sugar. It's an organic base bound to the ribose sugar with a phosphate group, adenosine monophosphate. And this one, it is a messenger. You know, we have cyclic AMP. We shall study it in coordination, but still I made videos about this. So this is a secondary messenger, second messenger in cells. And they do perform different functions. Okay? And they all require the ribose sugar. We have this RUBUP. Okay? Which is a carbon dioxide acceptor. Without this RUBUP, ribulose bisphosphate, then there is no way how you can respire. Can you imagine? You cannot respire. No. The plant cannot photosynthesize. And imagine if plants can't photosynthesize already. Even you can't exist. You cannot exist. Trust me. Okay? Yes. So really, these ribose sugars are, really good. Okay? Yes. So chemical tests for carbohydrates. So a moment. So chemical tests for carbohydrates, they are all reducing sugars, obviously. And so all of them being reducing sugars, when heated with a Benedictine solution, then what happens? The color of the Benedictine solution is going to change. Okay? From what to what? From blue. Now you need to know that's the order. From a blue solution, this is food tests. And this is food test. From blue solution, it is a solution. To a green solution. To a green solution. Okay? And then to a yellow PPT. Now yellow is a precipitate. Okay? Yellow precipitate. Write the whole word. And then to orange. Now depending on the amount. Yellow PPT. And finally to brown. Orange, there is E. To brown PPT. So depending on the amount, it will determine which color that you will have. If you have little, then you stop at green. If you have moderate, then you stop at yellow. If you have much, then you stop at brown. Okay? We stop at orange too much. Now, why this? Why this? Why changing the color? Now it is simple. When you heat, now the Benedictine solution, okay, is made up of copper 2 sulfate. And this copper 2 sulfate is blue in color. Now, since they are reducing agents, since all these monosaccharides are reducing agents, so when you react it with Benedictine solution, they will reduce the copper 2 sulfate, which is a blue color of the Benedictine solution. And copper 2 sulfate, the copper is copper 2. The oxidation state of copper is copper 2. Okay? And it will be reduced to copper 1. Okay? Which is formed as an
What's the
oxide.
color of copper one?
The color of copper 1 oxide is reddish brown. Okay? The color of copper 1 oxide is reddish brown, whereas the color of copper 2 oxide, it is dark. It's black, okay? But for copper 1 oxide, it is reddish brown. So now, depending on how much copper is reduced to copper 1 oxide, it will determine the color, okay, of the resultant solution that is formed. If the color is green, then it means just little copper 1 oxide has been formed. If the color is yellow, then somehow moderate. But if the color is reddish brown, then it means too much copper 1 oxide has been formed. And so that means there is too much reducing agent. Okay? And the reducing agent for it will be oxidized. Okay? That's why it is a reducing agent. I hope that one makes some little sense. Yes. What's the time? Okay. Today we have time. Now for disaccharides, just from the name, di means two, then saccharide, sugar. So di, that is two sugars. Okay? Sugars. So these are simple sugars, or these are sugars that are formed from condensation of two monosaccharides. Two monosaccharides combining together, they form what we call a disaccharide. And they are formed as a result of a condensation reaction, okay, between the two monosaccharides to form a 1,4 glycosidic bond. And it is this 1,4 glycosidic bond that keeps the two monosaccharides condensed together. Okay? We are going to see. There is nothing to worry about. So some examples of monosaccharides. Number one, you have maltose. And this one is formed from condensation of two glucose molecules. And commonly, it is found in germinating seeds and most cereals. Because most cereals, they store starch. And starch is formed by condensation of very many glucose molecules. Then we have sucrose. Now this is the sugar that you take at home. If you took breakfast, then this is you took the sucrose. If you put sugar in your tea, then you put sucrose. And the sucrose is also a disaccharide that is formed from condensation of a glucose molecule and fructose molecule. You have your glucose, okay, and the glucose condenses with fructose, okay. Then what is formed is the sucrose, and this is what came in your practical. They gave you this, and they want you to do some chemical tests. And you deserve some sticks. And it is found in cane sugar and roots of sugar beets, which are source of now what are sugar beets. I've never seen them, but what I'm sure of, the sugar cane is purely sucrose. Okay, and most of carbohydrates in plants are transported in the form of sucrose, in this format. So it is also a transport form. Then finally we have lactose, and lactose is made up of glucose and galactose. Okay, and this is the milk sugar. It is found in milk. Now the properties, I hope it's what is next. Yes, the properties. If they ask for the properties, just simple. Think about the sugar that you take, that you've just taken. Number one, is it soluble in water? Number 2, if it is soluble, is it sweet? Number 3, if it is sweet, is it powder or they are crystals? Number 4, does it pass through a selectively permeable membrane? No, they don't. Number 5. Number 5. What have we talked about? Yes. Is it reducing sugar? No, all of them they are non-reducing sugars except maltose. Okay? They are non-reducing sugars except maltose. So that means when they give you sucrose, like what we gave you, and you test for reducing sugars, you should get a negative test. But when you test for non-reducing sugar, then you get a positive test. Number 2, they give you lactose, the milk sugar, and you test for reducing sugars, you should get a negative test. But when you test for non-reducing sugar, then you get a positive test. Now when they give you maltose, okay, and you test for reducing sugars, then you get a positive test. That's why when they gave you starch and they say add there your saliva, and then you test for the reducing sugars, you were able to get the positive test. Why? The starch being a non, it is not even a sugar. It will be hydrolyzed. Now if you say broken down, I will cross you. So the starch will be hydrolyzed to form the maltose. Now when you come and test for this maltose, then that's when you get a positive test. Okay? Yes. So any queries as far as what we've covered is concerned? So basically that's how easy these disaccharides are. So let's see how they are formed and then we call it a day. So being disaccharides, we say that they are formed from condensation reaction. Now what is a condensation reaction? It is a reaction that proceeds with loss of a molecule. Now somebody might say a water molecule, not only water molecule, but as long as a molecule is lost. Now the molecule could be water, the molecule could be HCl, the molecule could be ammonia. So any molecule, okay? Any molecule that is lost, as long as there is a molecule that is lost in that reaction, then the reaction becomes a condensation reaction. Now similarly here, so you can see, let me get maybe which pen, maybe this one because it is not, it's what is not there. So you can see, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, this is carbon 5, and this one is carbon number 6. The same applies 1, 2, 3, 4, 5, 6. So the OH on carbon number 4 in one glucose molecule and the OH on carbon number 4, so the OH on carbon number 1 in another glucose molecule, they will approach each other. And so because oxygen, like I told you, has lone pairs to donate to one hydrogen to form water, which is now lost. Okay? And when it is lost, then it will form this. I think. So the oxygen is going to remain as a bridge that is bridging this glucose molecule to this glucose molecule. And so what is formed is a glycosidic bond. Let me write it here. Glycosidic bond. But because of the IU pack, then we need to indicate the positions of the glycosidic bond. And the positions, one is on 1 carbon atom, another one is on 4 carbon atom. So that's why it becomes 1,4 glycosidic bond. Is that fair? That's brilliant. Now for fructose, same thing. You have the OH, okay, and the OH. Now if this is your carbon 1, then this is carbon 2, carbon 3, carbon 4, carbon 5 and 6. If this is carbon 1, then this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and carbon 6. And we said we consider the lowest. The lowest is most favored. So they do condense to lose a water molecule, okay, and the product that is formed, okay. So you can see this one the same as this. No. So this is glucose and this one is fructose. So what is formed therefore, it is going to be 1,2 glycosidic glycosidic bond. And for galactose, it is the same thing. Well, the glucose molecule condenses with the galactose molecule. And what is formed? The 1,4 glycosidic bond and the disaccharide is going to be galactose. So I think we shall stop here for today. Okay? Go and read about the previous work.
Teacher,
Yes.
what's the functional group there?
Where?
In the fructose.
They are all alcohols. They are all OH groups. They are all OH groups. Okay? You can see these ones, these ones. They are all OH groups and they are the functional. Maybe you want to say this oxygen. No, you leave that oxygen. Yeah, so